Okay, so let's go ahead and go through the process of meiosis. What I want you guys to do is take a piece of paper and draw out a cell. You create the cell membrane, give it whatever shape, kind of arrangement you want, but it's going to be your cell membrane. Inside of it, I want you to create two pairs of chromosomes. So there's chromosome pair 1, and I'll show you its buddy in a second, and chromosome pair number 2, and I'll put its buddy here in a second. But color code them. So make the first chromosome of the pair one color, and the second chromosome of the pair another color. Now, the simple representation of this is, here's the chromosomes you inherited from mom for chromosome number 1. This is the one you inherited from dad for chromosome number 1. This came from mom for number 2, dad for number 2. Okay, so this is all going to be happening during meiosis 1. And we're going to walk through the general steps here. So at the end of meiosis 1, we're left with 2 cells. There's our 2 cells at the end of meiosis 1. So meiosis, supposed to be meiosis 1 here. Okay, so key things, prophase 1, and this is where definitely make sure you guys focus on this. During prophase 1, the chromosomes of the pairs have the possibility of crossing over. We don't know, it's pure chance. So let's say pair 1 crosses over at the top of the chromosome and at the bottom, but pair 2 only crosses over at the bottom. Pure chance. We don't know. Hard to say where they're going to cross over, how many will cross over, if they will even cross over. But that is a major event of prophase 1. This is what gives us our genetic diversity as sexually reproduced individuals, so crossing over. Okay, so the chromosomes appear, they cross over, nuclear envelope fragments, all that happens during prophase 1. They line up in metaphase, then they move into anaphase and they rip apart. Now this is the key. When they go into anaphase and they pull apart, the red chromosome is going to have purple tips on this side, or this top part and this top part. So we want to draw, here's the red chromosome, but it has, or I think it's called magenta, it has these little magenta tips to it because of crossing over. So that's going to give it some new genetic information. The other side of the chromosome, the magenta side, or the magenta chromosome, now has little red tips because it basically swapped tips with this chromosome. Okay, the red one over here, it only has one magenta tip, not 2. So those chromosomes are already different. The magenta chromosome on the left side here from pair number 2 is going to have 1 red tip. I said this is all pure chance. So anaphase 2, these chromosomes are separated away from each other, and what they're going to do is they're going to be separated, and eventually that cell will split in half. There's our contractile ring squeezing the cell in half, separating this chromosome for pair number 1 away from this one. This one for pair 2 is separated away from this one. Both chromosomes wind up going back into a nucleus within this cell here. There will be a nucleus in that cell there, and we will have pure chance now, random assortment. This is a theory of segregation and theory of independent assortment. We have no idea to know if this chromosome for pair 1 will wind up here. So let's just say by chance it does. Okay, so we got the magenta chromosome. I'll get 2 red tips on it in a second. And let's say by chance this chromosome winds up going into the same nucleus. So there's our red tips. It goes into the same nucleus as that magenta one there. That's independent assortment. Keep in mind what that is. Okay, so those two are in that cell, in that nucleus. And then down here, what we're going to have is the magenta chromosome with the one red tip, right there, and the red chromosome with two magenta tips. Down in this chromosome, or in this nucleus. Okay, so by chance, just pure independent assortment, these two chromosomes went into this cell, and these two wound up going into this cell. Next time, it could be the two chromosomes on the left in a cell, or the two on the right in a cell. That's why we have so much variability. Now imagine doing this with 23 pairs. That's why we have so many different possible combinations. Alright, now let's go to meiosis 2. So there's meiosis 1. Come all the way down here. We're going to call this meiosis 2. Each of these cells will divide, and I'm going to draw it big so I can fit it in there. Not representative of the actual size. The cells are actually getting smaller as I go through this. But after the cells take a break, that interkinesis, then they progress into meiosis 2. Chromosomes condense in prophase, they go into metaphase, anaphase, telophase, etc. But the key here, when they are going into anaphase, they rip apart. So we have this chromosome ripping apart, and it will rip apart. So you'll have a red chromatid, magenta chromatid, and then another magenta chromatid, but the magenta chromatid on the right has two red tips. That chromosome right there rips apart, and it has a full red chromatid, and then another chromatid with a magenta tip that separates. And anaphase, we're going to pinch the cell in half and start to separate it. Let's go down and do the same thing down below. The magenta chromosome separates. Magenta chromosome with the red tip separates away. The red chromosome, and then the red chromosome with the two magenta tips separate. And now they are chromatids. Anaphase, pull them apart, start cutting the cell in half, and now we have 4 cells. Okay, so here's the randomness again. You need one chromatid from that set and one chromatid from that set to combine and go here. The other chromatid from the set 1 and the other chromatid from set 2 go here. Same thing here. So there's set 1, set 2. So for simplicity, I'm going to take the 2 chromatids on the left and I'm going to move them into here. And then I'll take these 2 and move them down here. Okay, so we got a magenta and a magenta tipped with red on it in that cell. Down here we got a red, and then we have a magenta with 2 red tips like that. Come down here. This time I'll do the same thing. Let's take these 2, move them here, and let's take these 2 and move them down here. It doesn't have to go like that. It could change. It most likely will change. But this gives us magenta with 2 magenta tips on a red chromosome. And it gives us a red chromosome and then a magenta with a red tip down here. Like that. Okay, so apologize for the crude nature of the drawing. Sketch pad and a stylus isn't the easiest thing to work with. But what I want you guys to do is get the point and look at the fact that none of these cells are alike. The genetic information in each of these cells is different. So now I want you guys to pick a cell, A, B, C, D, and you tell me which of these is going to be the egg produced this month. Pick your letter. Whoever picked letter C, you're correct. It's pure chance. You get a 1 in 25 chance. So now this month, this was the genetic information passed in the egg. These 3 cells dissolved. Nothing worked. Nothing happened there. They're gone. They do not function anymore. They are reabsorbed into the body. They're what we call polar bodies. So in the meiosis process, in a female, oogenesis produces 1 egg, or we call it the oocyte, and generally we're going to say 3 polar bodies. The polar bodies, so they just dissolve, reabsorbed by the body, not used at all. That 1 cell is what potentially will become your offspring this month. So look at the genetic information. But what happens if next month A is the egg cell? Your offspring inherited different information. Some of it's the same. They inherited a big magenta chromosome, but this chromosome was primarily red with a magenta tip. That's different than red with 2 magenta tips. So on and so forth. That variability happens every single time a individual goes through meiosis. Now in the case of sperm, when we're dealing with sperm, instead of 1 egg and 3 polar bodies, when we have a male going through spermatogenesis, let me get rid of that, the individual will produce a sperm, give it a little tail, give it a little helmet, give this one a tail, another little helmet, another little helmet, little tail there, another helmet. But it's the same scenario. Which of these 4 cells, which of these 4 sperm cells will actually fertilize the egg? You pick. Let's see if anybody guesses right. I'm going with C again. This month, that sperm is the one that would fertilize the egg. So that's what your child inherited from the father with that fertilization event. Next time, if I said, well, let's change it up, let's get rid of C, and next time let's go with B, child just inherited different information. Okay, so oogenesis, egg production, and 3 polar bodies. Spermatogenesis, sperm production, 4 sperm. They're all genetically different, and that's what I want you guys to remember. Each of these cells is going to be genetically different than each other. And then every time we go through this, we're producing genetically unique cells. Okay? All right. So let me save this. And yes. Let me wrap up meiosis lecture here.